Good morning, everybody.
Good morning,
Good
sir.
morning, sir.
Good morning, doctor. Good
Good
morning.
morning for all the colleagues in Sri Lanka.
Good morning, sir.
Good morning, Dr. Gauri. Good morning, Shanaka.
Good morning, Dr. Shanika.
Yeah, good morning.
So, are you working from office now or from home still?
Dr. Vibha, I'm working from the FA office today.
Okay.
But we can only have two in the office. So in the conference room, only myself. In the office, just two people.
Okay.
Yeah,
Yeah.
the portraits on the back wall are very beautiful.
Yeah.
So this
FAO
is
ones.
so you are in Colombo.
I'm actually in Colombo during the week, and Monday and Friday going back to Kandy.
Okay.
It looks like we have 14 participants.
Shalini sent a message.
Yeah, I think so. I think so. Let's start, and there will be more participants joining as we get going.
Good morning, everyone. Thank you for joining us. So we will start today's session with a recap of day 2. So we request Dr. Vibhavja to give a small recap on yesterday's session. Please, Doctor.
So very good morning to all. Today is our third technical session, and as you all know that we are very systematically trying to cover every phase of the testing pathway. Our day 1, of course, was, as I indicated yesterday, the context setting day, where we had presentations on the technology, on the broad paradigm, on how the approach is, and how to collect the baseline information through the biology document. So yesterday we had gone through the phase 1 and the phase 2 testing very nicely by Dr. Tyagi. Through the phase 1 testing is done in laboratories or cages, and then phase 2 was the initiation of the field testing, that is the confined field trials. And in between we also learnt about the planning the field trials, how to do the what work or what considerations should we follow when we start planning our trial in terms of the information collection and other things. And then kind of through this journey of testing, of course, with our objective of seeking the approval, we have to continuously generate the data, and how we should plan that was our supreme Now, moving on from there, today what we will do, we will first have a presentation by Sir, by Dr. Tyagi on the phase 3 testing, and then followed by phase 4 by myself. And before we move on to risk communication, I have also included a small presentation on risk management considerations, giving a comprehensive view. So we will have that, and then the risk communication, and finally how should you need to interact with the regulator, how you have to compile the information and submit to the regulator. All the hard work that would be done in all these testings will ultimately go to the regulator for seeking the approval, and the final release. And the regulators would be basing their risk assessment on the information that you provide, and that is what would be in the last presentation. So we move on from here. We had Dr. Naina to ask a few questions, but the organizers have informed that he will be joining a little later. So we'll have a detailed question answer at the end of the presentations. So now we will start with the presentation on phase 3 testing by Dr. V.K. Tyagi. Over to you, sir.
Thank you very much, Dr. Vibha. I'll take just 50 seconds. Is it coming on the screen?
Yes, sir.
Is it fine? Hello?
Yeah, it's fine now.
Okay, thank you very much. As Dr. Vibha has announced, we will today discuss and interact for discussion on the phase 3 testing of the staged field releases in the nature of the genetically modified mosquito. For example, Aedes aegypti, the vector for deadly dengue infection. We have so far understood that there is nothing like making a shortcut or jumping to a conclusion, and all the intervening steps are very important. The metrics of the information in the intervening steps is all very, very important. So we cannot jump from the knowledge of the mosquito Aedes aegypti in the initial stage to the final stage of the fourth stage of testing without actually understanding each and every aspect of the risk assessment and management related with the testing of the mosquito in different stages of the testing. And today, what we are discussing is basically an extension of the ecological setting of our testing in the field in the stage 2. But this one is an extension of that with a viewpoint to generate more and more epidemiological data, that is the GM mosquito, just as the opposite strain of the wild Aedes aegypti is also capable to transmit the infection in the same intensity as we had been finding with the natural populations of Aedes aegypti. Incidentally, though we have seen this slide earlier yesterday, here the point is that there is a proof of concept that the GM mosquito is a good technology to reduce the vector population and thereby also impacting the reduction in the cases of the disease. Now, first of all, we need to understand why do we do the stage 3 of the field testing? What is its role in the evaluation of the GM mosquitoes? Releases in the environment play a critical role in the evaluation of genetically modified mosquitoes, particularly for the assessment of mating competitiveness, which is one of the most singular factor or study needed to be done in extenso under this stage. The longevity as well as the dispersal, because these affect a lot in order to generate uh, risk assessment features or characteristics, and for determining the efficacy of the intervention in an environmental setting typical of the insect species, that is the wild nature of Aedes aegypti. Releases also facilitate the collection of information for biosafety evaluation. This is very important, that without coming to the stage 3, we did not have a peep into the feature of the accumulation of information which could be used for evaluating the biosafety related measures like risk assessment or risk management of the problem emerging out of that. Now, as we know from the presentation of our Sri Lankan colleagues and from the data worldwide, that by now several open field releases of genetically modified mosquito have been conducted and published in the scientific literature. The study done in Brazil has been very extensive in generating the information, and I will refer to this study quite often during my presentation. Now, one of the most important and foremost and paramount responsibility in the stage 3 testing is the site selection. Unless we develop the parameters or information to select a site, nothing can move in this particular phased study. So first of all, what do we need? We need to make all the informations available to us. Information of the climate, information of the environment, information of the demography, information of the dengue cases for past 10 years, the information about the mosquito, its wilderness in the nature. We make a long list, and then the concerned scientists or the managers pay visits to the sites of many areas and then narrow down to make a very short list of the areas or the sites which fulfill our criteria of conducting the experiment. Finally, after we monitor the biology of the vector in nature in that site over several months and in all seasons of a year, then we confirm to that particular site which is meeting many of our criteria, which I will be discussing in my following slides. So we need to have certain features, certain criteria, certain ideas to go for selecting the site in the field, and these criteria are an important consideration and will be largely dependent on trial objectives and design. The main considerations, however, are what are our scientific and technical requirements, that is the objectives of the research. These may differ from one technology to another technology. The community engagement and ethical consideration, which are the integral part of the overall stage 3 conduction of the experiment. Without having the favor of the community or the stakeholders, we cannot succeed in this stage 3. And of course, without re-emphasis, the importance of approval or acceptability by the regulators or the regulatory bodies. The regulatory body must be able to give permission to carry out this stage 3 only then this stage can be proceeded further. Now each of these will need be tailored to the context of trials objectives. So these are general features of the overall matrix of characteristics of site selection. Nevertheless, these may vary from one particular technology to another technology to undergo. There is a prerequisite associated with the site selection. The open release trials can only be performed after a complete risk assessment has been performed and the efficacy of genetically modified mosquito has been ascertained in the contained trials. Now, this must be borne in mind that the stage 3 is not simply conducting a scientific experiment. As I said in the very beginning, that in order to make an analysis of the risks involved in the GMM mosquito, we need to start the risk assessment right in the beginning, right at the first stage of the testing, and through all the testings. And this will be culminating in the stage 3 of the testing. And without having made a complete risk assessment, the stage 3 testing cannot be accomplished. So what are the selection criteria for selecting a trial site? There must be people living, because we know that in nature mosquito is very intimately associated with the living of the human beings. It's highly anthropogenic. The mosquito Aedes aegypti on which the test has to be made must be naturally occurring as a dominant mosquito species in that area. The area must be also having history of the cases. As I said in my previous presentation, that all these features are important together by the time we complete our second ecologically settled experimentation. We need to be also generating information about all these kinds of essential criteria about conducting the field trial. After selecting, we set our objectives, or we already have the objectives with us, and we see that there is a consonance in our objectives and the selection of the site for investigation. 3 important studies need to be invariably conducted out, whatever technology we use, and these 3 are mating competitiveness of the GM mosquito with the wild population, the longevity and dispersal, because there is a inter-border movement. Inter-border movement means the area under your selection and the area around it. So there would be a possible dispersal of the mosquito from outside to inside or inside to outside, and therefore we need to make what is the rate of the mosquito which is traveling in to out or out to in. And the third one is for determining the efficacy of the intervention in an environmental setting typical of the insect species. For example, if already there are measures being taken by the state or the national body, what is the survival rate of the insect under those impacts? And unless we have a check on these things, performance of our GM mosquito will be always very precarious. It will be very confusing whether the reduction is by our GM mosquito or by the other control methods which were in vogue in the area. The releases finally will also facilitate the collection of information for biosafety evaluation. So we will continuously having the data generation on the risk assessment and how to conduct risk management if there is a some kind of a disturbing result. there has to be a very careful consideration born in the mind when we go for pointing out or selecting our site for experimentation, knowing that the mosquito can travel anything between 200 to 500 meter. We need to see that the site of these experiments or phase 3 studies should not be less than at least 200 meters. That is what the mosquito would normally travel, and with the help of the wind current or wind support, it can travel even more than 200. But nevertheless, we need to see that our sites of the studies are not radially closer by less than 200 meter in any case. Better it is kept at 400. The experimental sites and the control sites both have to be chosen for the phase 3 trials. I have taken this slide courtesy my friend in one of the institutes here in India, where the sites were primarily selected to investigate with the wild population of Aedes aegypti. And this is how the 4 sites were selected, which were about 400 to 500 meters apart, both between the control to experimental site and experimental to experimental, as well as control to control site. The distance is adequate enough not to not allow Aedes aegypti, the GM Aedes aegypti, to mix up with the population of the other site. As I have told you that when the experiments are done, there is always a danger or the possibility of the mosquitoes dispersal accidentally or by taking the assistance of the wind current over to other areas. To prevent this, hundreds of OV traps are fixed in different peripheries, and which sees the escaping mosquito and the adult mosquito and the future progenies of that mosquitoes. They're all trapped in the 2 different kinds of traps called BG sentinel trap for adults and the ovi traps for trapping the gravid females before oviposition or for their eggs. These are those BG sentinel trap. For the adult, you see that that is what on the top position, left position, and the ovi traps, which are used to trap the gravid female. The ovipositions of the gravid females is on the right side. And hundreds of them are fixed on the periphery of the villages or of the site, urban or rural, and these prevent any escape of the adult mosquitoes from the experimental site. The collection of the ovipositions or collection of the egg into the ovi traps, when it is compared with that of the wild before the site was selected, would show whether we were able to really catch or trap the out-moving mosquitoes or not. With this graph, we see that over several weeks and months, the experiment have generated that any mosquito which had been moving out from this area over to other had been trapped into this gravid trap and the sentinel traps, and this shows the significance of these traps. Now, there are certain key issues associated with the transgenic Aedes aegypti release in open field, and these are to be very, very strictly observed because some of them are uh, they are very challenging, they are very disturbing as well. Number one is that there is always a sense of uncertainty on the release of ratio of the GMM to the wild population mosquitoes. This has to be ascertained uh, by using mathematical models because in Brazil, experiments performed in Iragaga urban area, it was found out that the ratio used was 54 GM mosquito to one wild population. Now, this comes to roughly 0.03 or 3%, that is, out of 100 GM mosquito, only 3 wild mosquitoes, which later on, because this was found in 2014, and in 2016, the ratio was reduced to 0.13. That means even less than 3 in 100, that is 1.5 or something out of 100. This would mean that depending upon the site selected, depending upon the objective of the experiment, depending upon the mosquito technology being used, our selection of the release quantum of the mosquito will also differ. And therefore this uncertainty is always one major factor to choose what number needed to be periodically released, because if you over release, this creates a nuisance. If you if you under release, this damages the very central fiber of the control program by GM. Number two, another mosquito which we had been simultaneously talking about along with Aedes aegypti is Aedes albopictus. If Aedes aegypti is theoretically reduced by the GM technology in the nature, it is observed that Aedes albopictus, another vector for dengue and a very potential vector for dengue, which is paralleled now with Aedes aegypti potential for transmitting dengue, also emerges very fast. So these features are to be observed, are to be kept in mind when our technology is brought to the field. And one very disturbing result was seen in Thailand, where Aedes aegypti was reduced substantially, not substantially, moderately, and it was found out that after certain period of time, dengue or dengue hemorrhagic fever, which is the deadly syndrome of dengue, emerged with a vengeance. So reducing the mosquito population only moderately will not serve the purpose. The GM mosquito, though it is claimed that in Brazil and Cayman Island, the mosquito population were reduced anywhere between 85 to 95%. We need to see that this is sustained for a longer time, because moderate reduction may not give us the desired benefit. So what is the risk assessment associated with the stage 3 testings? The risk assessment associated with the selection for open releases should consider the isolation of the site in terms of its structure and the knowledge of the vector population, the disease dynamics, and the implication of any differential impacts, that is, the factors like climate and environment and other predators and chemical control programs going on in that area. When selecting the site, risk assessment could make use of the substantial advances in technology and knowledge of the geographical surveys. Now, this is very important. We need to select the site not only by physically on ground sighting the site or visiting the site, but we need to take in consideration the help from the GPS, the GIS, as well as high resolution satellite imageries when we are selecting a site. This will help us to select not only a site in a physical sense, but this will be fulfilling all our criteria for making out the experiment with the objectives intact. These methodological advances allow thorough analysis of temporally and spatially referenced data, both in time and in space. Whatever data had been collected, this will all be used for selecting the site and in consonance with the biology and the ecology of the mosquito in reference. This also includes choice of the appropriate site. This is what I said, that size of the site is important. If we take a unduly very large size, the entire pressure will be on the laboratory for production of the large number of the egg batches, larvae, pupae, sexing out the pupae at that stage, and then production of males and females. Now, this is very important because everything should be determined in consonance with our own potential present in our laboratory. If our laboratory is able to produce about 1 million mosquito on daily basis, we cannot take an experiment where we do require 2 million of the mosquitoes on daily basis. Of course, the periodic releases of the mosquito involve the weekly or fortnightly release of the mosquito. But what is important here is the selection of the size of the site should be done in consonance with our potential to produce our mosquitoes for release in our laboratories. As Dr. Weber was telling, that we need to also consider sometimes the cluster size and the number of our experiment because this would give us comparable data, both with the comparator with the species and other species in the region of the testing. And such a cluster study needed to be done over various and many different seasons, as well as possibly many years. After we have made all the observations related to risk assessment, and that will continue throughout, we need to keep also the management part of the risk estimation. The risk management in phase 3 is nothing but an extension or in continuation to what we had seen in phase 2. But this will be at a large scale now because our area has grown extensively and our objectives have also grown extensively in terms of the volume. The evaluation of surveillance data would benefit from the availability of appropriate baselines before release, such as the level and seasonal pattern of the disease burden, the past level of the vector population, effects of conventional vector control methods, which were all discussed in our slide when we were discussing about the criteria for selection of the site. A recall or control plan of sufficient scale to limit the species should be agreed upon and be available before we make any final analysis or final risk management studies. This is important because unless we take into consideration what other impressing or compelling impacts for control of the vector were prevailing in that area, we would not be able to chastise our risk management factors. At a minimum, of course, an additional risk management measure would be to stop the genetically modified releases with the very first information. If there is something going wrong with the experiment and our risk assessment has given an inkling that something is going wrong, we must take a decision to not go further. And that is why the risk management is very important, carried out hand in glove with the risk assessment investigation. In such a case, a more extensive and intensive conventional control capacity may be required to eliminate any residual population of GMMs after release. For example, if we find out something is going wrong with the GMO mosquito, we need to immediately press into action the conventional control methodology in the area to see that not a single seed of GM mosquito is left in the. This is emergency plan. This is very important, and we cannot always guarantee that a GM technology will be giving absolutely compatible environment when it is released in the field. So the risk assessment and risk management have a inexorable necessity to be conducted in the phase 3 experiment or phase 3 studies. The RM plans should draw on the results of the RA. That is, RA was always to be conducted right from the very first initiative in our experiments, and the RM must always look at what is RA producing or generating, and RM is the information on to decide whether the experiments are giving substantially good results or not. The effects on the biological diversity of the experiment, whether gene is transferable vertically or horizontally. In very recent paper in 2019, it had been found out that the RIDL gene was also found in the other natural mosquito population. Though it was at a very, very low ebb, nevertheless, the knowledge or the information is important, that in contrast to previous understanding that the gene cannot be transferred to other mosquitoes, now we are seeing that gene is transferable. Let me also tell you that this is not the first instance of the gene transfer in the nature from one organism to another organism. We had documented already that the invertebrate genes can be transferred to the vertebrate genes, but then this is always transposon based situations, and the technology used in the GM mosquito is such that the possibility is greatly reduced for any transfer of the gene from the GM mosquito over to other mosquito population. Nevertheless, it is very important for the generation of the data for RA and RM that we keep an eye on what could, even in the remotest sense, happen in order to feel ourselves totally strengthened and prepared. Now, finally, in a couple of slides, I would be completing my talk by stressing upon the need that all through we need to also have a sense of attracting the attention of all the possible ingredients or components of our ecosystem of the experiment. We know that there are a large number of the paradigms, large number of the factors which are contributory to the onset and onslaught of the vector-borne disease in a given area, and therefore it is very important to communicate very intimately, very extensively, proactively with all the stakeholders in the study area. this subject of communication and public engagement is extremely important, which we will be discussing in our subsequent interaction a little while later. Nevertheless, this is just to tell you that unless we build up the communication and public engagement into our GM mosquito experiment at the phase 3 level, let me tell you, nothing will succeed. And finally, with the note of the hope that newer technologies are developing innovative methodologies to control the wild population in nature. And in a recent experiment in Brazil, a water-soluble capsules having the eggs of the genetically modified mosquitoes reduced the uh, wild population by as much as 95%. So we are hopeful and we are sure for the success of our efforts. Thank you very, very much.
Thank you so much, Professor Thagi, for giving us for presenting a very informative presentation. Now we would like to invite Dr. Vibha, our international consultant, to present on phase 4 testing, post-implementation surveillance. Please, madam.
My slides are visible. My slides
Yes,
are
visible. Yes, it is visible.
Okay. Thank you. Thank you, Dr. Tyagi, for setting the tone of, testing, in fact, setting the tone for a complete paradigm of the testing up to phase 3, which is the staged open releases, and all the key points that one has to keep in mind when one moves on to the wider scales and how the sites should be selected and so on and so forth. The fourth component, in continuation to what Sir said, is the post-implementation surveillance. Now here, uh, just a minute. So here, what we are doing is that we have done the staged open releases on multiple sites maybe, and then we have to now work on a large scale. Now the scale of operations will depend, of course, on the nature of the technology, based on the considerations which Dr. Tyagi already explained. So application of GM mosquitoes on a larger scale, on a wide area would be based on the results of the phase 3 testing. So remember the go, no-go criteria. If everything is moving fine, then we keep moving. If there's a problem somewhere, then we go back and do the analysis again. The decision to include as part of the national or regional program for vector and disease control by authorities. So once you have the positive decision from the authorities, then only the phase 4 would be initiated. Now the risk assessment for phase 4, in addition to all the considerations which are explained, an important component here is the surveillance plan. And this surveillance plan has to be put in place for the monitoring of the GM mosquito effect. And this includes predicting the likely manifestation of any potential resistance also. You're doing your efficacy, you're doing your all, parameters, but in addition to this, the likely manifestation of any potential resistance should also be included, and the risk evaluation will be set against the benefits of GM mosquitoes in improving human health. So remember what we said initially, that in the initial phases we are looking at efficacy of our technology, but subsequently we are going to also see whether we are actually reducing the rate of incidence of the disease also. So these are the phases where you would be sort of shifting more towards the epidemiological outcomes. The results of the risk analysis here, the complete risk analysis done for this, will be taken into account to make a decision on whether and how to allow the large-scale mosquito deployment and whether to adopt it as a component of the National Disease Control Program. So this is a very important phase, because after that, you are sort of get going. So surveillance will be like a kind of an ongoing activity throughout the trial phase, and this will be to assess the effectiveness under the operational conditions, both entomological and epidemiological, accompanied by the monitoring of the safety over time and under diverse situations. Long-term surveillance is, this is considered equivalent to, if we have to keep in our mind, when we approve new drugs, this is followed by, for a certain defined period, what we call a pharmacovigilance program. In this pharmacovigilance program, in medicine, these are very well established parameters where we actually see that the usage of drugs is there, and any kind of the sort of effect is monitored in terms of any side effects or any kind of problems, if at all they can come. But in case of GM mosquitoes, what we are looking at is not only the efficacy point of view, but also the environmental safety issues will also be taken care. The ongoing monitoring will help will be aimed at ensuring sustained quality and performance for disease control. See, please connect it with what Dr. Tyagi just now said, that it has to be a sustained effort. Halfway effort can, will not solve the problem and it may aggravate. So we need a sustained quality and performance for disease control, and determining whether any changes are needed in the management of either the GM mosquito technology itself or other aspects of an integrated control program. The surveillance plan is designed and implemented to detect movement and integration of the genetic construct also, within the vector populations, and detecting any unintended changes in the vector biology that may result in changes in the biological fitness, adverse changes in the vectorial capacity, and changes in the nuisance impacts. In case of failure to perform as expected, things can happen the way the may not happen the way we want. The emergency control or mitigation measures should also be available to eliminate the escaped or established GM mosquitoes. Like any other public health intervention, GM mosquitoes will also require ongoing monitoring to determine whether their efficacy has diminished with time or because of the unexpected effect that become evident when used in new areas. Appropriate measurement of the entomological outcomes that guided the deployment of the GM mosquitoes actually must be continued after the trials also cease, and the type of depending on the technology, the deployment strategy, multi-year follow-up may also be required. So at the phase 4, when we are at the phase 4, we have already undergone a lot of extensive efficacy testing. The behavior in natural settings has also been established by the phase 3 activities. In spite of that, you need surveillance because it cannot be assumed that it will continue to behave as expected. Now, by analogy with the implementation of insecticides used for long-lasting insecticide-treated bed nets, residual spraying, and larviciding, efficacy can change due to changes in the genetic constitution or external factors such as weather and human activities. But the intervention at this point is no longer experiment, but is a control program whose ongoing effectiveness is being determined. And therefore, we will monitor a subset of the epidemiological outcomes that were utilized during phase 3 to determine whether these positive effects are being sustained. It is also likely that the GM mosquitoes which are deployed over large areas, only longitudinal passive clinical surveillance would be practical. However, in case a loss of efficacy is noticed, similar to the appearance of the insecticide resistance, we might have to go for any second generation GM mosquitoes, but those second generation GM mosquitoes cannot be deployed immediately, saying that, oh, I have this backup. So that backup will again have to be tested from phase 1 to 3 and monitored in phase 4. So what we have also noticed in case of, particularly for drugs, people always keep a kind of a backup event. So you have a one lead event and a backup event. So you can actually test the backup event in parallel throughout the process. In the new drug discovery, this is the approach people follow. So this is for the developers, that you can always consider having a backup events which can be, sort of parallelly tested up to a certain level, so that in case you feel any problem, then you have another second generation to introduce. Now these are all the observations which are in our guidelines, and as I said, it is very difficult to give a kind of a very defined checklist kind of thing for surveillance plan, because of the diversity of the approaches. Some are population suppression, some are population replacement, and so on. And I encourage the developers as well as the assessors to look at for any further guidance on these things. Go back to the WHO guidance and then clarify your doubts, because we have prepared all these things based on this consensus document, but in a much simplified manner, so that it is like a implementable and easy understanding for everyone. So this is all about the phase 4, not too much of details, but the basic considerations which need to be there. And as I said, it will depend on case by case. So now I want to share with you a little. Dr. Tyagi touched upon the uh, topic of risk management a little bit, but risk management considerations have to be throughout. So, and what we are discussing now, in part as part of our training, as part of the national biosafety framework, is the biosafety compliance. And for the biosafety compliance, the risk management is very important. So I have, although it is not in the topic, but I've included about few slides, and I would like to share that information with you. So just give me a moment and I will shift to that particular slide deck. So is it visible? Can you? No?
No, still you are there on the screen.
Okay, I will just do it again, sir. Just give me a moment.
Now it is coming.
Thank you, sir. So this is a little bit of an important component of the whole, guidance process, is the risk management and the development of an emergency plan. Because time and again we talked about the emergency plan should be in place. So we thought we will just touch upon this information also. So here, Dr. Tyagi already explained to you that risk analysis has three components: risk assessment, risk management, and risk communication. Risk assessment is science-based, risk management policy-based, and risk communication is the interactive exchange. So we are now going to talk about a little about the risk management. So risk management basically means that we have to identify and implement measures that can be expected to reduce risk to an acceptable level. Risk of doing these testings of a novel product which is still under development. Which development and testing. Risk management encompasses strategies developed to avoid and reduce risk to acceptable levels. It should identify and evaluate proportionate measures that are needed to mitigate any harm or uncertainty. It demonstrates how both standard and responsive measures would make the identified risks acceptable to the regulators. Now, I think sir also mentioned this very elaborately, that risk assessment and risk management both should focus on the particular GM mosquito application under examination, and its objectives will commensurate with the phase of testing we are in. I will explain you how it will vary with each phase. So the risk management plans would be prepared to appropriate for any potential resistance to the genetic modification, and then measures to mitigate harm or uncertainty, or the wider receiving environment also is very important. Risk management of the GM mosquitoes requires the development of risk frameworks in which scientific evidence is used to assess the probability that an adverse event will occur and the extent of harmful consequences associated with and without mitigation, if we have not taken care or if we take care. Now, at different testing phases, as I said, the risk management considerations will vary. And what is the basis? The basis is the exposure levels. So the level of exposure is much less in contained trial than the open releases. And then with sterile versus self-sustaining. You see, this is where the case by case will come. The first is, of course, as we move from the phase 1 to phase 4, the management considerations will vary. The second is the type of your technology. If it's a, remember the first slide when I said that the population suppression versus population replacement or self-sustaining versus the ones which have to be introduced, the risk assessment perspective is different, and so is the risk management consideration. So at each level of testing, from laboratory through the field trial, we have to ensure that safety, and to quantify or provide a qualitative rank or of risks associated with the eventual deployment of GMMs. Mitigation plans should focus on how necessary it is to obtain absolute separation in order to achieve safety and efficacy endpoints in the trials/release. There is no room for mistake here. We have to try to ensure, at least in the beginning, that the exposure, all the mitigation measures, that this can be taken out. It is very simple in case of plants. It's a simple field trial. We make sure that it doesn't persist. By post harvest, we will be having on the experience where it is. But then in this case, it is like you have to try to have a mitigation plan. We cannot allow them to escape without without the proper measures. So if you look at the phase 1, which is the laboratory or a cage testing, here my risk management measures for human health would include ensuring that the GM mosquito colonies and feed sources are free of human pathogens. So these are the practical things that you have to take care. Ensuring that laboratory staff is not carrying mosquito transmissible diseases, limiting unintended biting opportunities to guard against disease transmission by preventing and removing mosquitoes flying outside cages, and by ensuring that laboratory staff wears suitable protective clothing. Risk management, the second component would be to respond to the escapes from the laboratory. Escape detection system and standby mosquito control capacity sufficient to control adults within the dispersal range of the mosquitoes, and or conducting experiments in seasons when the adult dispersion and mosquito larval sites will be limited. You have already been informed by Dr. Tyagi in the phase 1, phase 2, phase 3 on how to plan the experiments. But these are the risk management measures that you have to keep in mind while designing your facilities, as also the study designs. Both are very important. When it comes to the confined field trials, and actually these slides are also a little bit of a recap, so that's why I thought I will include and share with you. The risk management measures in the field trials will include restricted access, clear and well managed standard operating procedures, the need for which I stressed in the beginning should be, and also Dr. Tyagi explained that every stage you should have proper SOPs, should be used to mitigate risks associated with the field testing. So applicants will be required to put in place SOPs to document how the material will move, the protocols to ensure site security, cage suitability, criteria for release strategies and surveillance, and then post-trial removal of material and cages. All these things you should have very good SOPs. Monitoring the performance of the containment confined measures will minimize the risk from the unintended releases, and will, as you move, if you make mistakes in the beginning, the regulator will not give you permissions further ahead. And if you want your technology to move ahead, these are the compliance to these measures is very, very important. And let me tell you, with my experience working with the developers, it is not difficult. Of course, Dr. Tyagi will tell specific things about mosquitoes, but we have seen in all this thing, if you have proper SOPs, if you have informed all your staff, that becomes a way of working, and that's what we should try to achieve at. Periodic sampling of the GM mosquito population in the trial should be undertaken to determine the stability of the transgene and any recognizable change in the genetics of the population that may affect the impact of the technology. A mechanism for practical and reliable discrimination of GM mosquitoes and wild mosquitoes should be available prior to initiating phase 2 experiments. For example, through the use of fluorescent dyes or dust or phenotypic or genetic markers. You should have a system of differentiation or different, detection. Where, where the release of male-only GM mosquitoes is part of the system, methods for reliable sex selection prior to release will be necessary to ensure an acceptable sex ratio is achieved. All this Dr. Tyagi explained about the sex ratios which are going to be released. You need to have a good system in place. Plans would need to indicate how the residual populations in the cages would be eliminated after a trial. So this is all about management of the trials. In the case that risk is determined to be negligible, this might simply involve allowing the material to enter the decomposer food chain. So it's not necessary, these are all big, don't consider them to be big words, but integrate them into you have to think about it. Once my experiment is complete, where does all the material go? So I have to think about it. Aggressive risk management of residual dead material would need to be considered. Then we move to the phase 3. Now here it is 1 and then 1 plus x for this one, and then like that, the basic things remain as it is, and then we are talking of the additional things. Risk management, as you are expanding your trials, you are moving from maybe a one confined field trial to the staged open field releases, you have to expand your risk management also to account for the lack of confinement. Now your confinement, first you were very having a physical separation. Please recall what Dr. Tyagi showed nice pictures of a lab, with fully protected lab, and then you have something in the open second stage, but then you are moving further. So your confinement is getting reduced and you are more into the open environment. So risk management will also have to be expanded along with that. Availability of the appropriate baselines are required before the release. We have already mentioned about it, but then it's again once again reiterating the level and seasonal pattern of the disease burden, past levels of the vector population, effect of the conventional vector control systems. All this baseline information should be ready. A recall or a control plan of sufficient scale to limit spread should be agreed upon and be available before field release. Without this, the regulator is not going to give you the permission. If there is an ongoing concern about the risk, there should be a procedure to monitor any degradation of efficacy in the GM mosquito control system that may indicate that resistance to the effector has developed. The degree of resistance, its rate of increase, possible hazards must be evaluated. I just mentioned all this above. In the staged open fold releases, you can also consider regular sampling of the wild populations as a method of detecting the resistance. Management should be put in place to avoid and detect movement through transport in case neighboring countries have not approved release for testing. Of course, in Sri Lanka, this won't be very relevant because you are an island country, but this is a very important consideration which needs to be kept in mind. Released GM mosquitoes should carry markers that ensure discrimination from wild release. Once again, same as in phase 2, staff should be trained about the risks of moving living specimens and should observe the transport protocols when moving any material. Post-trial monitoring should take into account the number of GM mosquitoes that were released with the aim of achieving an appropriate level of sampling efficiency. The post-implementation, the one which we discussed just now, here the risk management would incorporate appropriately designed surveillance procedures to allow effective risk mitigation. They must allow when it becomes impractical to maintain active surveillance as the GM mosquitoes become ubiquitous under self-sustaining there might be a plan in a way where we cannot observe. That's it, and it is living fine. So at that time we will be stopping risk, and we have to inform the regulator that we are going to do this only for X period, because after that it will be very difficult to monitor. Risk management should include any additional case-specific surveillance methods to monitor transgene activity within the GM mosquitoes that were required by the risk assessment, and they should also have options such as halting the releases in case of self-limiting mosquitoes, maintaining public access to the conventional disease vector control systems. You see, we do not know, these are all new technologies. If by chance it fails at some stage, we should maintain the public access. Don't stop those methods, they should be available. Designing stooping or recall mechanisms into the technology, such as greater insecticide susceptibility than present in the local mosquitoes. So these are some of the things, additional things which I thought will be very useful for the biosafety point of view, from the risk and management point of view of your trial, to satisfy the regulators that this is how we are moving, and also giving a clue to the regulators that these are some important parameters which need to be taken care while you are granting the permission. The emergency response plan should always be there at every stage. It should be like a documented, maybe 1 pager, 2 pager. It should provide safety measures and procedures for protection of human, plant, animal health, the environment and biological diversity against harms or damage caused directly or indirectly by LMOs or products of such organisms, as well as all. This is the requirement by the regulator, and all. Therefore, the emergency plan in case of mosquitoes would be in place to eliminate any escaped GM mosquitoes through proven means such as pesticide applications, insect trappings, etc. So having an emergency plan at every stage is very, very important, and this is what all accounts for the risk management side. And then we will move on to Dr. Tyagi now for the risk communication, which is our third important part for moving successfully for the conduct of the trials. Thank you, sir.
Now we request Dr. Tyagi to present on communication strategies application.
Yeah, thank you. I'll just take 1 minute to upload my slides. Are you able to see my slides?
Yes.
All right,
Yes,
I'll make it slide presentation. Is it okay?
Yes, sir.
Thank you. Well, thank you very much, Dr. Vibha. You simplified a great deal, not only my previous speech, but also gave an impetus for the risk communication, my current speech. Thank you very much. I also admire, let me take a half minute. I also admire our Sri Lankan colleagues for their great enthusiasm and energy. They are so good and great in sticking to the computers and interacting with us so enthusiastically. Thank you very, very much, everybody. So, am I audible? Am I audible? Okay. Okay. This speech will deal with a very, very important subject of risk communication. As I referred in my last slide in the preceding lecture, that without having made a very effective uh, communication with all the components of the ecosystem, it would be almost impossible to get through the entire process of success of the GM mosquito technology. And therefore, this is a very dynamic subject. It is to vary from one technology to another technology depending upon the objectives of the methodology involved. However, the principles of the communication which relate to determine the risk assessment and risk management analysis during the entire course of the investigation through phase 1 to through phase 2, phase 4. This will be very, very importantly centrally staged, and the component that I would be discussing would figure in every kind of a test, whether with one technology or another technology. So let me introduce by telling that majority of the public have a negative perception about the modern biotechnology, particularly the GM technologies. There is a very big tsunami about the GM technologies rivalry in many countries, and including also in India. Way back in 1970, there was a big uproar about the seed technology, and this has to be curtailed. And curtailing this negative perception is possible only through a very effective risk communication. Unless this risk communication is very effectively made, we will discuss what are those factors which make it effective communication later on. But unless we do this exercise, none of the technologies will see the sun finally. So this risk communication plan has to be strategically built up. It is not a casual exercise. It is not a easy or simple exercise. This has to be strategically networked, strategically built up, strategically documented, strategically strengthened. It's a great teamwork, and the most important components of such a strategy will be to have always a well-defined statement of the objectives of every GM technology before us, whatever we are undertaking into the field. Identification of target audiences and their information needs. It is not important what I like. It is most important what my audience like. It is the audience which has to give a green signal, a final green card to my technology, rather than I force my technology upon them. proposed activities to achieve the objectives. How we are going to get our objectives, it has to be well defined. What are our resources? How would we receive them? How would we network with them? How we would channelize them has to be very effectively understood and documented. And what are the means to assess impact and measure our success? Simply receiving the compatibility answer in the community for our experiment would not guarantee for the success of the experiment. We need to assess step by step continually our various experiments, their impacts, and their statistically measured success rate in order to be successful eventually. So first of all, as I said in my previous slide, the first responsibility of the team conducting the experiment and want to see the GM technology through, it has to construct, formulate its campaign objectives. The overall goals, which could differ from one technology to another, but nevertheless, the basic principle campaign objectives will remain like this. We need to build greater public awareness and transparency. Please do remember, there is nothing to be hidden, nothing to be feared, nothing to be worried about. Everything of the experiment has to be on the broadsheet. Educate audiences from time to time on impact of research. And this must be also born in our mind that we need to start this communication right on the first day onward. This exercise is not like a class teaching. This exercise is virtually imparting your knowledge by interacting with the people, by convincing about the scientific methodology involved to the people at different levels of knowledge, at different strata of the society. And therefore, we need to be very, very good. I will tell you how good the scientists or how bad the scientists are in communication later on. But what are those things to educate the audiences? Our academic excellence. That means we should be without any doubt about our work. There must be no ambiguity. There must be no dual thoughts. Whatever we are doing, we should know perfectly, completely about it. The institutional or company's integrity. Whatever we do, we are doing it with a perfect objective. Nothing hidden, nothing under the carpet. Everything before the people in a transparent manner. The experiments, as far as possible, should also generate employment opportunities for the locals, because this will impart some kind of a confidence into the local generation or local people that this thing is made for them. And finally, what is important is the knowledge to the people that ultimately this kind of experiment or technology will enhance their own quality of life. This will make them disease free. This will make them less diseased. This will make them sleep happily. This will have their family spending their time in the outdoors more calmly and without any vexation. So what is the objectives that we set before us? Let us inform people about our work. Provide a mechanism for public to voice their concern. If somebody want to ask any question, provide a meeting, provide a platform, provide a time for them to voice their concern. Do not say no tomorrow, no nothing today, no nothing. You need to always be so perceptible, so susceptible. you need to be very sensitive to their concern, and allow them to voice it. If you allow them to voice this, it will come very humbly, it will come very politely, and you will be able to think about an answer, a good answer to it. You need to increase mutual trust and develop the credibility with your other components of the community and ecosystem. Do not conflict or fall into the controversies of any kind, and if there is, try to reduce it by being very transparent, straightforward, and with your knowledge, clarify people who are doubting you or who are casting some kind of a doubt on you, or casting innuendos on your work. Discuss with them straightforwardly and reduce that kind of a controversy then and there. So, in your ecosystem, among your community, you see there are many people. There are teachers, there are students, there are laborers, there are doctors, there are scientists, there are businessmen. There are many people around you, but you need to target what are your audience. People who are highly educated and who are knowing the science a little bit, or they know about what is going on in the country because of their contact with the media. They ask questions which are more related to your science, to your doings. But there are large number of the people in your ecosystem, in your society, in your community, which do not know much about your science, which do not know what exactly you are going to do with your GM mosquitoes. They will ask you a question which may not be related with the science that you do. And mind you, as the great scientist Ronald Ross, the man who discovered the connection between mosquito and malaria, used to say that the best question ever to be asked is by a person who doesn't know about your science. If you answer that question, you really know your science and you really satisfy your questioner. So be very careful, be very knowledgeable. You need to also fragment or assort your audiences in the in terms of to build up priorities, because who will be making the largest segment of the community, which will have more doubts about or questions about your technology, and therefore develop kind of the segmentation of your audience in a way that you know from where the most difficult questions will be heard on you, and from where the questions related to your science will come onto you. So you need to be prepared for both of them, rather than to be only thinking that what you know, everybody knows that, without your telling them what you know. So the objectives can be scientific, but the common man, the components in your society does not know. So how do you convince them that what are your objectives? If you tell that I use this gene or this transfer, this transposon, this thing, that thing, they might not be interested at all into that. What they will be interested in, whether this particular thing that you are doing with the mosquito will cause any kind of a damage in the future generation or population or not, which is to be understood scientifically in a way that if a gene which you have introduced in a mosquito, if that mosquito is biting a person, whether the saliva is having that gene resulting into a change into the antigenic behavior of the saliva, and then when the mosquito bites a person, whether the person bitten will be having some kind of a severe allergy, yes or no. So one has to be very clear about the science of his technology, and do not hide anything. Each target group or audience will respond to different formats and styles of presentation and access information through preferred channels. Please do remember that we do not talk in the language, in the style, in the format which is Greek and Latin to my local population. We need to speak in the most simple manner. I have myself conducted a lot of such committee meetings on the roadsides in many cities like Chennai, and we have seen that they come forward to help in every possible way if they understand when you talk them in their own language, in their own way, in their own style. So please be a part of the community rather than impose your thoughts on them. The information link with the community or with your stakeholder has to be built through the right manners, through the right channels. The research community will always go for peer-reviewed journals. Publishing good scientific paper in good journal, international journal, is one very good way to garner the international support. You need to also develop a special website, which is a very good way nowadays to advertise your doings and confabulating in the scientific congresses worldwide so as to spread the knowledge generated out of your experiment. In the similar way, you need to also connect with the business community because the whole thing is finally going to be traded, and you need to also link with the community because unless you communicate through the local media like TV, radio, popular press, school and university lectures, internet communication, etc., the things will not go really worldwide. But there are different concerns also, like the consumer, the end user, will always think whether the gene technology is safe, whether the gene introduced will not come to me or will not come to my generation, and it will not give rise to some kind of a adverse or unwanted activity in my body. So all these things which are related to consumer, the policymakers, the teachers, the health professionals, the environmentalists in particular, we need to always get ready to answer their questions. Everything should be documented, nothing should be verbal alone. Anything that we are going to discuss with the various ingredients of our ecosystem, of our community, of our state or the country, everything should be supported by a document. Everything should be available on the paper. Everything should be available on the media in a way that it is easily comprehended by them. The language used, the style used, the format used should be one which they understand at different levels of the community strata. How it is to be achieved? Number one, by increasing positive media coverage. I need not tell about this particular thing in great detail because I'm sure that everyone now knows that media play a very, very important role in disseminating our viewpoint. And the geo mosquito, if we interact with the media and cover worldwide the scenario of our audiences, I think this adds a very great deal to our profit and benefit. Number two is we need to generate public support, that is the local people around us in the district, in the state, in the country, and this is done basically by carrying out credible, objective, factual information over to them. We need to make a dialogue, we need to make debate and participate into the public congresses. We need to make open communication no hidden strategies, no hidden agenda. And we need to discuss everything, the ins and outs, the benefits and the risks related to our technology, so and so forth. We need to be very transparent. This is one very important aspect of the GM Mosquito, that at any point one should not develop a kind of a doubt or some kind of a loss of confidence about the technology or about the company or about the institution carrying it out, both in its internal and external activities. Whatever strengths, whatever weaknesses, whatever threats we receive, whatever opportunities we are gaining, everything should be put on the paper, put in front, and should be easily available to anybody who want to see anything about our work. Make very strong partnership with the policymakers at both the state and the local and the country level governments, because they are the people to be depended on finally. And they may not know your subject very well, but they are the people who are to support you, and they are the people who want to come forward to understand. But therefore, what is important is kindly do discussions with them only in a way that they understand the matter, the central problem, the crux of the problem of the subject. So they should know everything in a way without any hindrance, without hiding anything, which is capable to convince them, and which is capable to attract whatever decision we want from them. We need to make several objects or several supporting documents, because these are important key factors to disseminate our doings. Strong visual identity, one-page fact sheet, press kit, brochures, video and slides, reports and other documentations, newsletter, news, stories, etc., of success and failures, in order to disseminate these wherever at whatever level these are required to be distributed. Now, this is the subject which is of greater importance, because scientists are very good people, but they may not be always very good skilled communicators. They may be very good in science, they may be very good researchers, but to communicate with the people around, people who are knowledgeable, people who are not knowledgeable much, people who are knowing absolutely nothing about your work, how to communicate with them, very, very few people are skilled in that. And therefore, you need to find out people who are the spokespersons and communicators on your behalf. In your company, in your team, in your researchers, among your researchers, you would always have some people who are very good scientists, but at the same time, they are very good spokesperson and who can define your things in a very logical manner to the people, that they get convinced about the subject of your doings as well as your outputs. Then it is very important to deliver your options to the endpoints, to the community people, by making information available to them. And the local culture and tradition should be always kept in mind. This is important because if you are doing a investigation of GM mosquito in a time when a whole village or whole area is engaged in some annual feature, annual function of highest significance in their life, what you will do? You will not get the local people working for you. You will not get anybody to talk about your subject with you. So you need to understand also the local culture, traditions, etc. of the local community, so that you can build up a very good delivery system of your information over to them. Your resource needs, though they may be paltry, they must be also known very critically to you. Your administrative structures, your secretarial support, your communications, travel, grants and material and supplies, all these materials which are needed continuously throughout and beyond your time period should be very rigorously and very in a business-like manner utilized, so that one does not fall short of the stipulated estimate of money or resources. Then the point is that there must be people, there must be scientists, there must be researchers who must develop very special expertise in the business of GM technology communication. Now, a strategic plan for public communication requires a variety of specialized expertise. This include communication specialists. As I told you, everybody is not a good communicator, but there are always certain people who can communicate wonderfully. Technical writers, that means people who understand the local cultures and local traditions. They write things including everything around their experiment designs in a way that it connects with the community very well. The graphic artist and illustrator showing the work or the approach to the work in a way that it becomes very pleasant. It becomes very elegant. It becomes very soothing to the people to whom we are making it for. And then we need to always continuously make the trainings to the people. And then how do we measure the success and assess the impact of our work? An initial assessment of public perception, as noted above, provides a benchmark against which to measure the impact of a public information initiative. The most direct kind of assessment is to survey a sample of people representative of target group before and after the initiative. This is the same group when I said we should segment our target audience. This we must make communication more extensively and continuously, because the valid results depend on a properly structured questionnaire and survey process. As I said, the local language, the local tradition and cultures, and the mood of the people, all those things had to be carefully observed when we structure our questionnaires. And at any time, we must be very strict and stringent to review our processes, our steps of success. We must question ourselves, like the 6 W's. We must always ask ourselves what, when, who, where, etc. What should we continue doing? Whether we stop doing, whether we adjust, did we do what we set out in the plan? Did all our activities work toward the agreed objectives? And how many people did we reach? Etc. etc. These are the kinds of things which we must continue to confabulate with ourselves and among ourselves within our scientific community, as well as with those who are able to guide us. And then finally, we measure and evaluate our outcome as what is the end result of our activities. Did we achieve our business goals? How did we change our behavior? Did we contribute to win hearts and minds of our cause? Now that's the beauty of any risk communication, that it is not only the conduction of a research activity, but whether we have also won the hearts and minds of the people for whom we were doing all this great activity. The summary of the whole risk communication can be displayed in this manner. We need to make an audit of all our work. We need to set our objective very carefully in the beginning itself. The strategy of the plan, the communication network system that we need to make, we should be very clear about. And then what we finally got at the end by winning the heart and mind of the people to whom we were working for. Everything should be designed and kept in front for everyone to see and follow. And thank you very, very much.
Thank you so much. So now we request Bajaj to present on the information requirements to discuss. Please, ma'am.
Is it visible now?
Yes, yes.
Thank you, Dr. Tyagi, for a very nice and a systematic explaining a systematic approach to the risk communication. So with this, we sort of come to the end of the meeting the three requirements. But what we will do, we will quickly go over. I mean, these are all the considerations that you have to keep in mind. But in terms of operational, operational work, then you have to compile all the information and then submit it to the regulator for seeking the permission. So for that, there is a section at the end of the guidelines, which is an annexure and which indicates what is the information that is required for risk assessment. You see, all such things, whatever you do, are for you, but finally what you submit to the regulator is going to be the basis of the decision making for taking further your technology. So it is very, very important that the data that is submitted should be of very good quality. It should be focused. It should be only relevant data which is required for the risk assessment. So I'm just going to quickly go through this checklist. This won't take more than 5-10 minutes, and then we will open it up for discussions and further issues. So if you look at this, why have we prepared this checklist is basically as a useful reference, but it does not mean that, as I said right from the start of the workshop, that it has to be case by case. So particular risk assessment information requirements will also be case by case. So information listed here may not be required in all cases, and sometimes which is not listed here may also be required. So this is just an indicative checklist to give you a starting point, and then you have to see what is your product and where does it fit into. Okay, so the first of all, we must describe what is our mosquito, GM mosquito that we have developed, name of the GM mosquito, common name, scientific name, what is the trait we have introduced, origin or source of the genes. If there is a unique identifier number, we should put that. Intended use, are you applying for field trial or you are applying for release? Purpose of the modification, life history parameter, and the life stage which will be released. Description of the GM mosquito will continue with the pedigree map, which strain you picked up and how you have modified, complete thing, geographical areas within Sri Lanka to which distribution is intended, history of use in control programs, methods to distinguish the modified mosquito from a non-modified mosquito. Remember I mentioned about the risk management, that this is a basic requirement of the regulator. So molecular, morphological, or whatever method you have, that should be provided for. In addition to that, if the suppose it's not a locally developed but you have imported, then we should know what is the status of approval in the country of origin, status of trial in the countries, and how much quantity did you import and from where. The biology document was explained to you. The information on the description of non-modified mosquito is equally important, and that should be provided to the regulator, because remember, your regulatory agency does not consist of all mosquito experts. This is an interdisciplinary team. Somebody is expertise in molecular biology, somebody may be another subject, and so on. So all of them need to have this information, and as even the regulators have to be familiar with the basic information, only then we can move forward. Otherwise, you will be raising as regulators questions which won't be applicable to this particular scenario. So this is all the basic information which should be provided, and this can be taken from the biology documents or other peer-reviewed publications. Please do not include hearsay information ever. It should be very authenticated, because it can fall back on you. Your results may be different, or it may not be, authenticated. Description of the donor, each transgene which is present, each component, the complete information of that has to be provided, in the dossier or in the application. So description of the donor organisms, how you have modified, what is the characterization of the genetic material, and a summary diagram of the complete construct also has to be given. Molecular characterization should be done. All this work would be done in the labs, where exactly it has been inserted, insertion sites, identification of open reading frames, etc. Molecular biologists are very familiar with it, and all of you must have done all this. Again, once again, I say, it is not that everything is applicable to everyone. This is a checklist. Tick wherever it is applicable, provide the information. If it is not, explain why it is not, and move on. So confirmation of the intended effects, what has been expressed. Phenotypic characteristics of the GM mosquito should also be provided. Phenotypic stability of the inserted trait, life history parameters if there is any change, survivorship at each life stage, mating strategy, flight ability, oviposition rate, etc. Ability to persist in the environment. All these parameters are, are to be provided. Things like response to the detection survey tools, status of susceptibility to the labeled insecticides employed as a risk management option or for control of the wild type mosquitoes. See, all this has an implication in your risk analysis. Once we know these are the susceptible ones, then only we can, check whether you have properly included them in the risk management or emergency plan or the disposal of the residual, things. Then we've already discussed a lot about this, the site characteristics. I presented first, and then Dr. Tyagi very nicely explained each and every parameter today. So description of the confined field release site has to be provided as part of your submission. Description of the confinement measures that you are going to take. Is it going to be physical, ecological, or what would be your measures? Geographic isolation, if any, is already there. All that should be properly explained. The risk management plan, what you are going to have, method for monitoring, method and procedure for controlling in case of unexpected spread, plan for protecting human health and environment. All this will be part of your risk management plan. Now this is what all is in the application form. Now I am going to add a few general information to you. We had given this information in our first training also. For some of you it may be duplication, but even it is worth talking about it for all the new attendees, as well as even for the old ones, just to reiterate this point. The data must be of high scientific quality, yet sometimes it may be high scientific quality. Of course it is required, but if it is not relevant to the assessment, then it is of not much use for the risk assessors. So do not give irrelevant data also. Pick up do very high quality work, and then provide what is most relevant for your application. So I have to have a focused attention. Risk assessors, the people who are part of the regulatory committees, they must also determine the relevance of the data and of the data they collect themselves. See, assessors Also for their confirmation, take material from risk assessment reports on the biosafety clearinghouse, other resources, as also there are risk assessment reports by other countries. Data quality information from the national and international standards and guidelines, experts and other regulators may be used in the risk assessment. Using the high quality relevant data makes the risk assessment process simpler, easier, and results in a report that assists in the decision making process. You see, you want an expeditious decision, come up with a good application and a dossier. Collecting irrelevant, poor quality data needlessly prolongs the assessment process and hinders the decision making. You have added just for the sake of adding, and the regulator raises questions on that. Then you will be, lost in that jumble and will not be able to achieve your objective. Data transportability is the use of data to support a risk assessment for a particular country that may have been generated in another country. So you can always think about it. This is not a new concept. We are all, as scientists, used to referring to the research papers published in other countries. Scientists commonly use this by other researchers. We always move on from what has already been done. So same thing can be done here, but you have to see which is the appropriate data to transport. Data which is geographically independent. For example, something generated in the laboratory where standard methods have been used, data was collected under controlled condition, and the test materials were well characterized. Such data you can definitely use, and from the other countries, but you have to explain to the regulator that this is the reason. If I'm going to repeat it, it is going to give the same data on a case by case basis. So this is very common in all countries. This kind of data is accepted when it is in the controlled conditions. The key points to remember as far as your information requirements are concerned: developers need to focus on producing data, again I am repeating, that is relevant to the risk assessment. You have to do some out of box thinking to satisfy, as sir said, people on the street will have lots of questions. So document those questions which can be probable ones by the regulator or by the communicators or by the public, and then try to have, data doesn't mean all experiments. Data also means information collection from other peer reviewed publications and so on. It is not about filling a bucket with data and then thinking how it can be used. Instead, having a clear cut requirements in your mind. Sometimes there may be additional requirements by regulators, but it is very important for regulators also to think that they should give additional requirements on the basis of safety, whatever is their mandate, not just for the academic curiosity. We have seen this, oh, if you try to do this, let us see what happens. So that sort of thing should be avoided. It should be separate, need to know from versus nice to know, and requested information must be useful to decision making, and as I said, not simply to satisfy the academic curiosity. So with this, we close, and we would request if there are any questions and move on to the question answer phase. And for all the participants, let me tell you, I'm reiterating, all these slides would be compiled and will be submitted to NSF and to the project office for, as a training manual. And then in that comprehensive form, it can be circulated to all the participants.
Thank you so much, Doctor. Now it's open to the questions. We request all the participants to ask their questions if there's anything. Doctor, the comment that we forwarded, do you want to address it separately?
The comment about mosquito, or the comment that you, okay,
Yes.
those comments, yes. Those comments are for incorporation in the guidelines.
Yeah.
So we will, I had indicated, replied back to you that they are for changes in the text of the guidance. So now the thing is that guidance has once, some of them are, can be addressed, and some of them are already there, but we need to highlight and inform back to the scientist who has raised. But you know, the document has been already cleared by the LTO. So what we will do, we will see how best we can address. We will definitely address all these comments, and again maybe go back to the LTO and then tell him that these were the comments and some of which need to be addressed. So we will look into those comments. But my request is, you know, that see we had circulated this document, so it would have been really nice that if this came at that time. But nevertheless, I value all the comments which come. We still have time, and thankful to the scientist who has given these comments, and we will suitably address them. They're more related to the language in the guidelines.
Thank you so much, Dr. Thanks.
Right.
If there's any other questions or comments, please feel free to convey them. Professor Gunathilaka, if you are there.
Yes, Shalini, thank you very much for all participants joining this workshop for 3 days. So today is the last day of the workshop. I think the NSF has shared what the draft guidelines and how to test, carry out all these phases for GM mosquito testing. And if you have any concerns or suggestions for the guidelines, so it is better if you can express your suggestions or any other corrections.
Sure, you know, I have one suggestion that please give them particular time, like, you know, within a week or something, so that then we can address all those comments in a comprehensive way.
Yes, that's good, Dr. Vibha. Dr. Gauri, we have circulated this one before the meeting as well. And yesterday also you had circulated this guidelines to the all participants, the potential participants. Yeah, so we will remind them again that within, as Dr. Vibha said, for a week or whatever the duration, written comments are entertained during this period, right?
Right, okay.
So, Dr. Gauri, are you happy about the supplementary material I have provided now?
I couldn't go through it, Dr. Naina. I will go through it and let you know.
Yeah, it's just a summary of what the guideline says. Just a basic summary, like it contains about 7 pages, including the references. So, I mean, your objective was just to, without referring to the whole manual, to get an idea within a shorter period of time. So that covers the objectives as you requested. Yeah.
Can you share a copy of that with us also? Then we can include it in the training manual.
Yes, Dr. Ahuja, actually all for consultants, they are giving, we have requested all for consultants to give a supplementary document. Once we received, all the documents will be shared with you to incorporate, combine as a supplementary document, because Chanaka will explain little in detail. Chanaka.
Yeah, that's a plan, Dr. Vibha. So these are actually any additional information related to the country context. Because guidelines are, I mean, you know, when we are using the guidelines and anything that we know about the country, so that information we collected from the consultants, and that was the plan actually to share with you, so you can incorporate that into the training manual.
Okay, great. Yeah. Very good. Because, you know, because the participants are asking for presentations, no? So I thought sending these individual presentations, sometimes there is linkage is not there, no? Something which we speak. So that's why I want to give it in a comprehensive form to you. So if there's anything, whatever comes, you know, you can share that so that I can finalize that.
I have a question from Professor Tyagi. First of all, I want to thank both of you for the nice presentations. I would like to know more about the cross-species gene transfer. You mentioned about the gene can go to,
Yeah. Thank you for drawing this very important point. There has been an argument whether the gene to the host animal like mosquito, when it is released in the nature, whether there is a possibility that the gene either horizontally or vertically enter into some other kind of population of the type of the mosquito which is not our target mosquito, like the other species of the mosquito in the nature. Now one paper has recently come in which it had been very clearly and scientifically documented by molecular studies that the gene in the GM mosquito has been found in the other natural populations which were not targeted. Okay, but the argument behind this is this, that this is not a new thing. In past also it had been demonstrated that the gene from the invertebrates have also gone into the vertebrates, and though the pathway of the transfer from invertebrate to the vertebrate had been a different one, and the pathway, the way in which the gene is introduced into the GM mosquito is different. Therefore, the possibility from the GM mosquito to go into another kind of a species population is relatively negligible. But this is on record.
So do we consider that as horizontal or vertical? Like, is it?
No, this particular one is a vertical one, which I am giving to you. But because the horizontal transference of the gene is not yet proved. Like the lizard eating upon the mosquito is not going to be possible.
And one more point, Shanika, that one has to keep in mind is, even if, like, we say, the scientists never say, zero, but they will, like sir said, negligible. So,
Yes.
but even if this kind of a thing has happened or it happens, what are the consequences of this? So the risk assessment is based on a phenomena happening, and then what is the likely harm from there. So you have to analyze like that. So we very commonly hear about transfer of genes to the wild relatives. I'm giving you an example in crops. So for example, canola, the wild relatives of canola, which is Brassica napus, are very common in Canada. But and there is a possibility of the gene flow also. But then there's a complete analysis of, if any of you want, we can share those papers also. The thing is, what are the consequences of that transfer? Is it going to create anything wrong? It's a gene from another very commonly occurring soil bacterium. So even if that gene has gone in the wild population, unless we spray that with the herbicide, it is immaterial. So such gene transfers are very common, like sir said, in the
Yeah.
nature. These things keep on happening, but what
Yeah.
is the probability and what are the consequences? That is very important to understand, for everything.
Absolutely, absolutely true. This is a natural phenomenon. I mean, this is nothing which is endangering the life of either man or the environment quality. Nothing absolutely related with that. But then it is on record.
The reason I asked is, like, in one of our expert groups, there's this one expert asked the question, like, do we need to assess the risk for other mosquitoes in Sri Lanka? Like, if we have several endemic mosquitoes, like, because there's a biodiversity aspect that we need to kind of look at it. So that's why I want to kind of clarify that.
Yeah, Dr. Shaneka, this is one of the objectives of the research with the transgenic mosquitoes. One needs to study that aspect also, whether the gene is at any point of the development or release is able to get transferred into other population. There is always a point to be studied.
Yeah. Sir, in addition to this, you have to first see, does this mosquito, the Aedes aegypti, mate with those wild types? If it mates only, then there can be a transfer. If it does not mate, then even if there are 100 wild species present, there is not going to be any impact at all.
Yeah, there
That
are
is the
two.
most
I
important.
fully agreed, Dr. Viba, with you. The point here is many mosquitoes are cannibalistic on some other species. Like toxorhynchitis mosquito is cannibalistic on Aedes aegypti.
But Aedes
So,
aegypti is not, no, sir.
Aedes aegypti, like toxorhynchitis species, feeds on Aedes aegypti. The point
Yes.
here is the point, as you rightly said, unless there is a connecting another species of the mosquito with the GM mosquito, there is no need. But if there is a connecting link between the two mosquito species, like I'm telling you, one mosquito species preferably feeding on Aedes aegypti. So one needs to study that aspect.
Yeah. No, I'm saying, sir, the other way around. The Aedes aegypti does not eat any mosquitoes. It
No,
is
no, no, no,
so
no.
now we are
Yes,
modifying the Aedes aegypti. Eating,
yes.
eating means any protein or anything would be destroyed. No, that won't have any effect on the gene transfer. Actually, once we finish with the question answers, if you guys have time, then I have some few slides which we had a very senior professor from who is the member of the Brazilian Regulatory Committee had shared with us, and those slides are actually exactly these kind of issues and how the Brazilians dealt with it. So there is a small case study, but once let us finish with the question answers. I mean, we have the time, so if 10 minutes additional is required, I can share those with you.
Dr. Shanaka,
Thank
there
you.
is another issue also involved. There is the mosquito like Aedes albopictus. In the nature, it tries to sire Aedes aegypti, and the eggs are laid, but the eggs are not viable. So, are you trying to understand the evolutionary aspect is anything which is going out of my body is going to the nature, whether it is embryonated or not embryonated. So, two different species like Aedes aegypti and Aedes albopictus in nature also are trying to mate. They never mate successfully, but they mate. And the point is that that particular point of gene transfer has to be studied.
Okay. Thank you.
Thank you so much. So we have a request for Professor Tyagi to share the title of these recent papers showing gene transfer between other species of
Yeah, I will send to Dr. Viva, and she may possibly send to any number whichever she is trying to know here.
Okay, thank you so much. So do you have any other questions? If not,
I have another question to maybe Professor Naina or anybody from the mosquito group. So in the Sri Lankan context, if we suppress the Aedes aegypti, is there a possibility that Aedes albopictus can pick up and we get the opposite effect that we are looking for?
Of course, if you try to control, suppress one population, there will be a replacement in the population. So the rebounding effect. So if you try to suppress one, definitely the other one will get a chance to have a wider dissemination.
So in the urban area in
And
Sri Lanka, do we see more from albopictus? If you take Colombo and those areas?
No, now the albopictus usually prefer the vegetations for their breeding. So when you look at the breeding ecology, we cannot have a 100% shift to Aedes aegypti to albopictus in the urban setup. But in the rural setup, if the Aedes aegypti is going to be suppressed with this technological driven experiment. So albopictus can take another chance of breeding in the more in this environment.
So, Professor Nana, here I would like to share with you. Actually, that's where I thought I will share that case study with you. Actually, this was the issue extensively studied in Brazil when they
yeah,
tried to suppress this one. But the conclusion they came to, what they did was they did a survey of the area where the Aedes aegypti, the incidence was very low. There was very low breeding and hardly any breeding, and but Aedes albopictus was prevalent relatively higher. So what they concluded was that it is not necessary. You do not, this cannot be, this is like a perception that, the incidence, once we suppress this mosquito, the other will increase. But the data indicated to them, I don't have the full details of it, but the conclusion that is there in the risk assessment report by which was prepared by the Brazilian regulatory agency, it says that it is not going to happen. That it is not that if we suppress the Aedes aegypti, the Albopictus incidence will increase. There is no known, kind of paradigm,
Yeah.
or no
Yeah, that's
reason for
Dr.
this kind
Iba.
of.
Now, with Sri Lankan context, some of the scientists, like the same research group members, have found that Aedes albopictus is also a very efficient vector in certainly in Gampaha district, right? So in that case, the Brazil, that may be different because the vectorial capacity of the albopictus may be different. But in our case, the albopictus also having they are certain in certain areas, albopictus are the leaders for transmission of the dengue disease. Now, what we can suggest, now we have different alternatives. Now, the riddle mosquito for Aedes aegypti, and there's a Wolbachia strain coming from Australian strain Wolbachia, that is for Aedes aegypti, and we have developed Aedes albopictus from Sri Lankan scientists. So, if we can combine all these together as one, we can suppress all these potential vectors rather than targeting one particular species. So then there, of course, we can have an integrated vector control with different approaches, maybe from the modified vectors or others. So then this may not happen.
Right. Thank you, Professor Nani.
So another issue, another point they said was, does it compete for the same breeding places?
Yes, they compete. Yeah, they compete because we have done certain field trials that these they compete with this. Yeah.
So if they compete, then there is a likelihood.
Yeah. Always there's a species succession in the breeding space.
Okay.
Can I add a word? Not only that Aedes albopictus compete for the same breeding habitat of Aedes aegypti, it also removes Aedes aegypti after some time from that breeding habitat. It is an invasive species, more invasive compared to Aedes aegypti. So one has to be very careful about Aedes albopictus spread in a country.
Yeah, definitely, sir. That's true. Yes, I also agree with you. Yeah.
Thank you so much. So, is there any other questions from other participants? If so, please feel free to ask. Otherwise, we can go for
Dr. So actually, this is not a question. So this is a request. Then you said that so you can share that canola gene transfer into this wild type species where they have done the risk analysis.
Yeah, sure.
Yeah,
sure.
can you please send that?
Yes.
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 Okay, thank you.
Thank you. We don't have much questions from the participants. We can
The voice is cracking.
can you hear us now, Professor?
Yes, now it is okay.
Since we don't have questions from the participants end, I think we can go for Dr. Viba's presentation that she was suggesting.
All right.
Actually,
Yeah, let's go to the presentation. So that might be.
This is a case study, which Professor Paulo Andrade from Brazil, he had actually presented in Colombo in 2014 when, we did around that time it was approved. But I found the way they looked at the problem formulation and other things very interesting. So I just took out a few of them, and I thought that if the time permits, we can discuss it. I mean, it may be of some use or it may not be. But since I have this information, I thought we will have a look at it. So very quickly, I will just show you. So this is purely from websites, and also specific to the Brazilian situation. It's visible now?
Yes.
Okay.
Yeah, that's nice. Yeah.
Yeah, this is about the regulatory risk assessment. for commercial scale release of transgenic mosquitoes. You see, Professor Paulo is a very, very esteemed member of the CTN Bio, a very, a person who is on the other side, he was the one who prepared the risk assessment report. So once again, reiterating that the general principles, case by case, comparative, step by step, science based and biological risk. And here he has again, stressed that it is usually undertaken by a multidisciplinary team with molecular cell biology and this and this. So this is the steps, which are followed generally in the risk assessment. We are going to have this discussion in our risk analysis framework, where we you first do the context of the risk, which is also referred to as the problem formulation. And this would include the important parameters, history of safe use, familiarity, impacted organisms. Actually, I should start this way. The regulations, biology of the parental organism, your construct, the environment that you are going, which other organisms are likely to be impacted, like we had this discussion about albopictus, familiarity, history of use. For every identified hazard, you have to find harms and the likelihood, where I said, the negligible, this, that, what we were discussing. And then there is classification is done, followed by the this one. But I just want to drive your attention to the some of the actual points which they used. Since I have, what happens is when you are in looking for the documents from Brazil, most of them are in Portuguese. So Professor Andretta was very kindly had shared the report also at that time and his presentations. So I thought it would be useful. So regulatory framework, biology of the chemical, this is just the repetition of what we just had. But in terms of genetic construct, all the new proteins, knockouts, expression levels, biota which interacts, and then knowledge derived from the non-GM and so on. Now here comes the context. So in the problem formulation, what generally the regulator will ask is, what are our protection goals? So the law will generally say, if you see any act like your own biosafety act, it will say protection of the environment and human health. That's what we have to ensure. But they are too generic. So once you know that you are going to work with AHFTI, then you have to sort of narrow down this to your protection goals. And Aedes aegypti is only in urban areas within or near human dwellings. So the protection goals must be translated in the assessment endpoints existing in this special environment. So here they have said that Aedes aegypti is highly anthropophilic and transmits diseases only to a few human and non-human primates. And these non-human primates are not existing in the urban areas, and that is why this animal health issue will be sort of kept aside in terms of the risk assessment. It does not mate with the other native mosquitoes. I mean, you have to do it systematically to keep ruling out and then concentrating on the key issues. It doesn't mate with the other native mosquito species and essentially with no other mosquito. That was their observation. So no animal feeds. You will very common say, oh, this animal, there are a lot of animals we feed on the other, fish, all that feed on the mosquito. So there, what the food chain will be affected, and these kind of issues ecologists will raise. So, but the point which they have concluded in their risk assessment is that there is no animal which feeds exclusively on Aedes aegypti or on any mosquito species. So those which are feeding on them are also not any valued species. So that's not a worry. We are not worried about the lack of food. We are more worried about. So this is like a risk benefit analysis. So benefit is much more if you are able to control the dengue rather than having that the food for some not so valued species is just going away. As a consequence, there are no plausible assessment endpoints except the human being. We are most worried about the human being. So the environmental issues are sort of taken care. What are the issues for human beings? Keeping in mind, only the direct biological impact is considered in the risk assessment. Even key issues like allergenicity and toxicity, any new infectious agents transmitted by these are the important issues. For any new infectious agent to come, there was no scientific basis. But in spite of that, the precautionary principle will say that we should look into this. And how we are going to look into this is the allergenicity and potential toxicity. Humans don't eat mosquitoes. So what is the route? Through the bite of the female. So we have to keep in mind that we are releasing the males, and so males do not bite. However, the exposure may be extended if the lethal trait fails. So all this, issues, like what I'm trying to say, and when I saw his presentation and he made how they have come to, the analysis and the answering those questions through the data. So like, people will say, oh, they will survive beyond, this one. But the survival is only 2 to 4 days. Actually, it is less than 5 days, but 2 to 4 days is the mostly time. So that's why you have to, sort of answer the queries by the public, like sir said, the public perception and other things, and also to the regulator, how you should answer. So this is how, if you keep answering through your data, and then you will be at the end, you will have to see what is the likelihood and what is the consequence of that. So first step will be, is this going to occur? Very high, is expected to occur, could occur, could occur in some circumstances and may occur. But even if it occurs, then what is the consequence of it? Is it marginal, minor, great, or major? So likelihood of the exposure to the sizable amount of protein may be very low. Most proteins do not have allergic epitopes and do not bear similarities. So hence the likelihood of any harm is absolutely marginal. And this is the table. We are going to discuss this table in detail in our risk analysis framework. Anything, those risks, perceived risks from the protection goal to the assessment endpoint and to that risk, we need to finally come to our risk classification, which, most of the time, see, even in this, there is no zero. So you will be actually seeing what is the risk and what is the probability of that, likelihood and the consequences, and then the amount of harm will be estimated. So this is the way to, do. So once the likelihood has been done, proteins present in the saliva of that, the risk actually here demonstrated through the thing that this is a negligible, and the very low probability, marginal, and then the risk is negligible. So in the risk assessment, more than one hazard will be listed. Initially, your list of hazards should be very long. It will be long list, but then as you keep moving, these hazards, exposure, they won't materialize into risk. As I said, keep ruling out that, oh, this is not going to happen, this is not. So like that, you will have the full answers to the commonly expressed concerns, and that will provide you the pathway to harm. And I'm not going to go into this detail because we are going to discuss this in the risk analysis framework, and I will actually include this kind of a pathway to harm there so that you can explain. I would encourage all of you to attend that one-day session on the risk analysis framework. So like, for example, here they have done an example of a multi-step pathway to harm. So general hypothesis is that the transgene does not work properly and may be transferred to the local A. aegypti population, leading to new strains of mosquitoes with enhanced ability to transmit the disease. So this is the hypothesis for which they are trying to draw whether this risk is going to happen or not going to happen. So, I mean, on the face of it, all these things look that, oh, this can happen. But then when you look at, break it into steps, like genetic escape from conditional lethality, likely to be no. So then they say, like that, they keep moving from one step to another. I will share these slides, and maybe the experts will have a better understanding of it. And see, this is how they kept on, breaking this into it, and then no meaningful risk, no meaningful risk, and finally they concluded that this is an hypothetical risk, and this is not likely to happen. So I won't go spend too much time. We are already late, but I will share these slides with you all. Similarly, failure of the construct to kill the mosquitoes. The trait is not fully expressed, and it may warrant the survival of a small fraction then would these surviving GM mosquitoes differentially harm human beings? The answer was that likelihood of such survivors constitute a very small fraction and the potential harm is marginal, so therefore the risk is negligible. So this is the whole process, that they have gone about. So in fact, I was reading about this A. albopictus, where they said, occupation of ecological niche made vacant by the control of Aedes aegypti. Said in this case, it is not the presence but precisely the absence of the aegypti that may be a risk. So we are not seeing the risk of Aegypti being present, the modified one, but the absence. So this is what they said, like you said, it may be true for the Brazilian situation. You have to see for your country. So in their case, they have said, does A. albopictus compete for the same breeding places? They have said seldom. The A. albopictus spread in the Brazilian cities still free from A. aegypti? The answer is no. Are there any reports of such opportunistic niche occupation? The answer is no. So like this, because of these, they have said there seems to be no reason to believe that technology would have, they've said generally that would have more risks. So, if you go like this systematically based on the data, based on the additional information, you can actually, lead to very consistent results and ensure that, we are adopting, if at all, but a safe technology and with all the due measures. Thank you very much. This I just picked up a few slides, but I will share the complete slide deck with you, Professor Naina, for your use if it is of some information to you. Thank you.
Thank you very much, Dr. Iba. Thank you very much for sharing this information with us.
Shall we conclude the session, Dr. then?
Yeah. So since there are no more questions, we will share this whatever the guidelines we prepared to the other participants, and we will get their feedback within a week of time. I would like to thank Professor Tyagi and Dr. Vibha for allocating your valuable time for this activity for 3 days. And it was really good experience and revised our knowledge as well, and nice preparing nice presentations and interactive. It was an interactive session, not just a lecture. It was an interactive session. So thank you very much. And I would like to thank the consultant for this component in SF and the FAO for organizing this event. Thank you.
Thank you. It was a great pleasure interacting with you all. Thank you very much.
Yeah, welcome, sir.
Thank you, Dr. Ahuja and Professor Tyagi. Thank you.
Thank you.
Thank you.